Okay, great. So good morning, everyone, and welcome to today's session of the Structure Meets Function webinar series. So before we start, I would like to introduce Instruct for those of you who don't know what it is. So, well, Instruct is a pan-European distributed research infrastructure which provides to the scientific community access to high-end technologies and methods in structural biology. So as you can see here in the map, currently it has 17 members and there are 11 centers providing access to its service. And actually, I work in one of them, the CMB-CSIC from Madrid, where is this Cryo-ET facility, and I work here as a computer scientist. So, well, let's go to the talks then. I'm gonna stop sharing. And well, our first speaker is Jiri Novacek. He's the head of the cryo electron microscopy and tomography core facility of CEITEC, another center of Instruct, placed in Brno. And he's going to give a talk on the platform for scientific data management he and his team have developed, called SIP. He will tell us about its main functionalities and how the implementation was in a mid-size Cryo-ET laboratory. So, Jirka, now you can start sharing your screen.
All right, thank you. Thanks a lot, Irena. So can you see the slide? Just briefly confirm and then
yes.
Yeah. All right. So thanks for joining. And even though this is a Structure Meets Function seminar series, we'll this time start with a topic that is more technical, and I'll still hope that there will be some structure, but not the structural data in there. So I'd like to present and show to you the platform that we have developed to facilitate the management of the data that are generated in a mid-size EM facility like ours. This topic is in general, I believe, relevant to those of you who are operating the service labs that are not connected to the large infrastructures such as synchrotrons. So when we produce the data for a user in our lab, we typically collect the data that are then shared somehow with the user, who is actually the owner of the data, and they will analyze their data, produce the results, which in a case of the cryo-ET microscopy will be cryo-EM maps that they will deposit to the database together with the coordinates that they will deposit to PDB through one depth maybe, and that is what they eventually need for publishing. Eventually, they can also publish the raw data following the publication of the results of the data analysis in the database that is focused on a raw data. In the case of the EM, this is Empire. Yet this is not a condition that is generally requested, and since there is additional workload related to that, this does not necessarily always happen. As you can see from this graph, where currently, or at this was done last week. So last week there was almost 40,000 EM maps available in the EMDB, which is the data analysis results repository, whereas that there is less than 2,000 entries in the Empire available. One can argue that for instance, particularly for EM, one data set can generate multiple structures. That is, of course, true, and the case for some, but the ratio here is significantly different and probably does not correspond to solely this fact. Similarly, and then I will now show you the data that was shown to me 3 years ago by Jose Maria Carrazzo. Similarly, during the SARS-CoV pandemics, the EM community has produced large number of structures, but only for very few of them the original data were published together with the structural data itself. So what we are trying to suggest and implement is the slight adjustment to that original data workflow, and that contains additional parts that allow us to preserve and transfer or hand over the data to everyone who needs the data in a more or in an easier way. So we are working with a cloud platform to which we actually upload the data directly from our instruments. Those data are made available to the user, to the owner, such that they can analyze them and then again publish the results. And we have developed tools that allow us publish the data or the metadata and the links to the original data upon either user request or upon the expiration of the embargo period. Primarily, we are not, in our case, working with a database that is dedicated for EM data, which is Empire, but that is something that I'll show in the end as an outlook, and we are using a more general database for the scientific data that is available. So, in brief, what do we need to do when we operate a facility and collect the cryo-EM data, or how does the specific cycle of the cryo-EM data look? As with any other imaging data, the cryo-EM data are typically very heavy on the size of the original raw data, because we're acquiring images, and per data set we usually have something like 1 to 2 terabytes of the raw images. So this is produced by an electron microscope. Then ideally we would have a computational infrastructure near the microscope that would allow us to do the real-time analysis of the data, such that we can get a feedback directly whether the quality of that data is sufficient or indicates indication that the data quality is good, and subsequently transfer the data to the storage through which we can make the data discoverable by the owner. Eventually, this poses yet another need for the facilities to have not only the instrumentation, but also a particular infrastructure for the data processing and data storage. So with that in mind, we set a couple goals when trying to develop an alternative system that would leverage the benefits of the fact that there are potentially other experts in the computation and data storage. But most importantly, we thought that the system that we want to implement needs to be easy to set up and easy to operate. It should not be platform independent, and it shall be also hardware independent. Of course, the hardware would influence the performance. We eventually thought of the workflow in a general way, such that it can be used not only to the cryo-EM data, that is our field of interest, but to any kind of data coming from different research infrastructures. But the cryo-EM data pose a typical or one domain that is very heavy on the size or size of the initial data, and thanks to that, it is one of those cases which might require extra attention, and managing those should show that handling other types of data that are not that heavy or sizey might actually be easier and manageable with the same system as well. So what we want to do for the EM data in particular is that we want to do the data annotation directly on the instrument, and then deployment of the data directly from the microscope to the certain cloud, and automate the archival and publication processes. Since we want to get rid of the necessity of running the real-time data analysis on our resources, we decided to join with the HPC center and carry out the data there. So here is the graphical display of the system that we've developed and we are now using. So we have our microscopes that are in the internal network, not accessible from the internet, and within that network we have a single instrument that has a secured connection to the particle web server. The web server is an orchestrator of the whole system and communicates with individual components, providing tasks to be carried out by that particular component. So the first step is a data transfer from the microscope. The data is transferred to the cloud provider. So we use IROTS, Federated Cloud Solution, as a platform to transfer the data around in our system. And once the data are transferred to this cloud storage that is close to the HPC, we can start the data analysis. For that, we've implemented two solutions. One is based on Scipion, and the other on CryoSpark. Both things were very popular in the community to run the real-time data analysis. Once the data analysis is finished, or directly at the start of the experiments, the links or the way how to get to the data are shared with the user via email, and the user can either start working on the same HPC if they have access there, or download the data from there and continue the data analysis on their own. Eventually, we use the EU-DAT services such as BPIT and B2Share to assign a persistent identifier to each data set. So with this, each data set is assigned with a PID or DOI, and then it can be made publicly available, or particularly the metadata that we collect are made publicly available through the B2Share. So now for some details how this works, and I will go quickly through it. As mentioned, everything is orchestrated via a single web server. to which the user identifies through a third-party identity provider. So we use either OrcID for the academic users or Google for the non-academic users. And that web server provides an interface to the graphical user interface that you can use to interact with the whole system. That web server also in parallel is an orchestrator and communicates with all of the nodes. Such a node might be either storage infrastructure, such a cloud storage, or the computational infrastructure, such as the computational resources, or the front end computational resources on the HPC, or actually the service lab. So the EM lab from which we collect the micrographs that are then submitted to the system. The web server can actually single web server can actually operate multiple labs. So that allows us to scale up and facilitate the utilization of the computational resources, because we can have multiple labs like ours connected to the single web server and utilizing the same storage and computational resources. A bit of a detail how this works and what are the requirements. So the only requirement from the network perspective is that each of the nodes that we call lymph nodes needs to have HTTP access to the web server, to the orchestrator, and the communication is only one way from the node to the server. The individual nodes are then handled by polling. As for the data itself, so the different nodes, the different lymph nodes have different roles. In this particular case, we have one node that actually pulls the data from the microscopes and pushes it, transfer it to the storage. We've implemented different type of storages. So one can use the standard physical storage or storage that you can mount to your system and transfer the data via defining a particular path. And we've also worked with two different cloud solutions. The primary one is IROX that we primarily use, and the secondary that we've tested earlier but abandoned later on was one data. And here is how this actually looks. So we are operating in Brno, which is one city in Czech Republic, where we have the Cryo-EM lab. And the data is actually transferred to the HPC center, which is in Ostrava at the IT4I. I mean, physically, this is 170 kilometers, but in practice, what's more important is that there is a good network connection between the two nodes, and including the last miles, as this is called, the network connection from the microscope to the HPC is always 10 gigabit or better. The spine network in our case is 40 gigabit. So the utilization of these resources removes the necessity of us having the computational infrastructure here. And as mentioned before, we could easily connect multiple centers like ours to this single infrastructure as well. minimizing the needs of having the additional computational resources scattered across the labs. So this, I mean, even though this is not physically close, it allows us to perform on-the-fly data analysis, and we have two different workflows available. One for CryoSpark, where we use the standard CryoSpark live workflow, and the other for Scipion, that for which we offer a predefined workflow, but we also have an option to download new workflow that is validated from the workflow hub. So a user can eventually design her or his workflow that is then uploaded to the workflow hub and can be directly used during the data processing. Currently, we use a standard queuing system on that HPC without priority for particular priority for the facility, and for the EM data, we can show that we actually can easily transfer them between these two centers within the time that is in general below 2 seconds, as shown for these almost 20,000 movie images, bearing in mind that each of them has some 150 to 300 megabytes. This time allows us to actually perform the data analysis in real time. And with that also obtain or receive back the feedback referring on the data quality that allows us to either interfere with the instrument or not. So I think that I should speed up. So we actually differentiate data into 3 types. The ones that are main for archival, the second that are alive just for the one data analysis campaign, which is the acquisition and analysis of the data set, and the third, which are temporary and are removed directly once they're after they are created and analyzed. So eventually, we aim to archive the initial raw data and the metadata, the workflows that are used for the data processing, and eventually the results of the, of course, of the data processing. How individual nodes work. So each of these nodes is implemented in Python, so it can run on both Windows and Linux systems, and it is meant to operate continuously. So being a part of, for instance, a service task that is run on that particular instrument. They communicate with the orchestrator, and based on that communication, they spread individual tasks to either their resources or carry out the task on their own. So now more from the user perspective, what I will try to show you is that even though this is now set up for the EM facility, it can be easily changed to any other type of a facility. So the standard configuration is performed through a single configuration file. We use a YAML format. So it is a single YAML format that configures how the facility and all its resources are set up. So for each facility, we can define multiple instruments, and if needed, each instrument can have its own workflows. These workflows are particular workflows related to data analysis. So in the case of EM, this would be either single particle or tomography or electron diffraction, for instance. One can easily imagine that this could be just the data transfer. That is the simplest workflows, and that can be implemented for any type of the solution and facility. And this is how the front-end application looks. So it is web browser based, so which means that the user do not need to install any additional software. It is accessible directly from the microscope, and it is actually the single point how to transfer the data from the microscope to the user. With that, we enforce that at least some minimal metadata are filled in before the data can be transferred from the microscope, and thus we generate a minimal data model that goes together with the data. The data model that we are working with currently has 4 components. It is the information about the operator and the center, so about our lab and about the person who actually is responsible for acquiring those data. Then it has an information about the user, so the data owner. We also collect some very general information about the sample, and then the information about the data itself. Our data model for the data part is actually inspired or corresponds to the ISPy-B data model that is used at some synchrotrons for Cryo-EM data acquisition. Once we start the job, the data are submitted to the cloud and analyzed, and we are getting a report on the screen with the updated data status, which allows the user to check if the quality is sufficient or there is an intervention needed with the instrument. In parallel, the user is notified by an email, and within that email, she or he gets a link to the project webpage, and on that web page, we share the information about the data acquisition, so the setup of the data acquisition, the report which was available on the previous slide, and also the information how to access the data, whether that data has been selected for long-term preservation, so for archival. And if that is the case, also a button to submit or publish the data through the public database, the B2 or UDA databases in our case. Apart from these features, the data management lifecycle, we've during the time added additional apps to our system. That from which I would primarily mention one that I found very useful. But we also have an app for the sample storage, so sample management, the booking table, the visual inspection of the time plan for the particular instrument and particular date. And this is very specific to EM. We also keep the information about the autoloaders content for the autoloader systems, and the current status, history, and time for which the individual grid samples been on a stage. But the most important part of this additional apps is the remote access. So through this application, we allow the users to access the instruments remotely from their home. So I already mentioned that they identify against the system using their own identity, ORC ID primarily. And whenever they have a session planned in the planning board, they can access the particular instrument that they have a session or on. The remote access, for the remote access, we use the Apache Guacamole. That means that we need to have either VNC or RDP remote desktop available on that particular instrument. And that's it. Otherwise, the Guacamole runs in the browser, which means that you can access it directly either from any PC or from your mobile phone without the need of additional software installed. This allows the user to either set up the session remotely or check during the acquisition the status of the instrument. And of course, for the administrators or the facility people, it allows to fast access to the instrument at any time to intervene if there are particular problems. So I will conclude with that. So we have put together a workflow that facilitates the life cycle of the cryo-EM data. It's composed of the part that automates the annotation of the data. It's transferred to the cloud storage, on-the-fly data processing, and eventually publication of the raw data in a state together with the metadata in a state that is closer to what one could call a fair principles. Eventually, we would also like to use this information, the metadata that we have, and the data from the data processing to run a feedback loop. That is something that we are working on, and eventually also initiation of the deposition to the public databases. And with that, I would like to thank particularly Radek, who designed and is working on the whole application. And this is a collaboration with Martin Golasowski and Jan Martinovic from the IT4I. There's also a collaboration from Irene, Carlos Oscar, Carolina, and Jose Maria from the CNB in Madrid. And with that, I want to thank you for your attention.
Thank you very much, Erika. A super interesting talk, especially thinking that this can be used in other techniques apart from cryo-EM. So congratulations. So maybe we can have one or two questions. I don't know. I don't see any questions on the panel. If there is no questions, I will go for one. So, well, so the first one is, Erika, this is currently being used at CEITEC, right? So,
yeah.
Is okay. And one thing is related to the connectivity between the web server and the other nodes. It's correct that the other nodes doesn't need to have any open port, just to go to perform HTTP request to the web server, and with that will be all, or
yes. Yes. So yeah, it is only that port that would allow for the HTTP sending the HTTP requests. For the microscopes, in our case, we have all microscopes in one local network, and using one PC in there that we call support PC to actually perform all the requests from all the microscopes. So then these send the request through the support PC. Eventually, for such a closed networks, you can get along by having one restricted connection to the internet, and with that communicate with the orchestrator.
Okay, thank you very much, Jurka. So now we can move on to the second talk. Now we have Elda Bauda, whose postdoctorate is taking place at the University of Geneva. since almost a year. And she is also quite related to CEITEC, since she got an Instruct grant access to the EM facility and was there in an Instruct internship program. So her talk will cover the topic of her thesis, the ultrastructure of micromolecular assemblies contributing to bacterial spore resistance revealed by cryo-electron tomography. So let's go with Elda.
Yeah, can you hear me?
Yep.
Okay, great. I will try to share my screen.
Great.
So
now we can see your screen, Elda.
So let's go to window. Can you see the full screen?
Yes, we can.
Okay, great. Perfect. Thank you for having me to this Instruct webinar. So today I will present some of the results we obtained during my PhD thesis. So I did this PhD at the Institute of Structural Biology in Grenoble, as you mentioned. So my two PhD supervisors were Cécile Morlot, bacteriologist and structural biologist, and Christine Morisco, expert in cellular EM. And those two areas of expertise led to a joint project aiming at performing structural studies inside the bacterial cell. And this work would not have been possible without the strong collaboration with the CEITEC in Czech Republic and sponsored by both iNext and Instruct throughout my thesis. So this PhD journey was amazing, but it has now come to an end, and I am since December postdoc in Roby Loeff's lab and Andreas Bollen's lab at the University of Geneva. So we studied bacterial sporulation, which is a morphological differentiation process that leads to the formation of a spore. So we studied this process in Bacillus subtilis, which is a non-pathogenic bacterium, but it can also be found in important human pathogens such as Bacillus anthracis, the bioagent of anthrax, or Clostridium difficile, which cause a severe gut infection and it's an important nosocomial infection. So when those sporulating bacteria are submitted to an external stress, they differentiate into a dormant and resistant spore. So how does it work? So sporulation starts by the formation of an asymmetric septum, which divides the cell into two compartments. The small one is called the spore, and the large one is called the mother cell. So the DNA is duplicated, and one copy of the DNA will be transferred to the spore in order to protect the genetic material of the bacteria. So it takes several hours to the spore to mature inside the nurturing mother cell. And once the spore is finally matured, the mother cell will lyse and the spore will be released into the environment. As soon as favorable condition returns, so meaning that the stress stops, the spore will germinate and return to vegetative growth and repeating the cycle. So as I mentioned, it takes several hours for the cell to create this spore. And you can see here electron microscopy images of resubstituted sample that show different steps inside this cycle. So during my PhD project, we were particularly interested in the structures that confer resistance to those spores, such as the protective layers on the outer surface or the compaction of the DNA. So bacteria are particularly challenging to observe due to their small size. As a matter of comparison, here are fluorescence microscopy images. On the left, you would have a eukaryotic cell, and on the right, a single bacterial cell as a green dot. Therefore, cellular features during sporulation are not fully elucidated, and their nanometric dimensions require high-resolution observations, for instance, electron microscopy. However, Bacillus subtilis is still too thick to be directly imaged by transmission electron microscopy. During my PhD, we implemented several sectioning techniques in order to make the sample electron transparent. So we implemented different ultramicrotomy techniques, 3 of them. including free substitution, Tokoyasu, and Semovitz. So for sake of time, I won't discuss all of them today, but I'm more than happy to discuss the drawback and advantages of these techniques later on. As you may know, cryo-FIB-SEM emerged as the most conservative techniques for sectionings in the past decades. It stands for focused ion beam milling, monitored by scanning electron microscopy. I had the chance to benefit from a collaboration with a neighboring institute, the CEA. A core-room cryostation was installed on a Zeiss microscope that they have. And this core-room cryostation allows to work in cryo condition. And this cryo device was installed at the beginning of my PhD. And one aspect of my PhD was to develop protocols for the manufacturing of lamellae in cryo conditions. This process was far from being straightforward because it was actually a lab mainly of material scientists, and there were a few users actually doing cryo and biological applications. So the core-room cryostation had some stability issues, and it took a really long time to be able to make the first lamella. And actually, it took me about 9 months to get the first lamella that could show nicely a spore. So at that point, I really needed to start to see how things are done in a facility and get expertise advice. That's when I decided to ask for a collaboration in the frame of the Instruct internship program. I was looking for a facility that had access to a cryos, a Titan cryos, and also similar core-room cryostation next to their microscope. So that's how the collaboration started with the CEITEC. So I was extensively trained by Jana Moravkova and Jiri Novacek on those two devices, so the Cryos and the Versa 3D FIB-SEM microscope. And in only 2 months, we obtained really nice results, and I could go back to Grenoble and mill much more efficiently and be able to have a result much more efficiently. So I used a rather standard procedure for sample preparation, but what I would say is important in my case is the use of a highly dense bacterial suspension that allows to have a uniform layers of cell on the grid. So as you can see on these SEM images and FIB point of view images, I really have like a carpet of cells covering the square, and this allows to mill a lamella actually in every grid square. I'm not limited by the position of the sample. So on the two microscopes I was using during my thesis, so the Zeiss and the Versa 3D, it was possible to do some kind of automation. On the Zeiss, I could do the rough milling, meaning that I could do the big trenches overnight, for instance, or over the afternoon. But the polishing, so the final step that allows you to reach this 200 nanometer thickness, had to be done manually. And this was really the limiting step at that time, because you have this problem of redeposition, meaning that you can't leave your lamella too long in the microscope. So you have to be fast to do the polishing. Otherwise, you will have like redeposition of material that will basically bring back a thick layer of ice on your lamella. So I was able at that time to do between 10 and 18 lamella per day on the best days, and managed to reach 150 to 250 nanometer thickness. What's nice with those bacterial cells is that, and this thick suspension that I have, is that I have plenty of cells in one lamella. So I have bigger chance to have my sporulation events, or even to have different views of the spore inside one lamella. So the lamella is then transferred to a powerful cryo-electron microscope for tomography acquisition. And an image, as you may know, an image is taken at each tilt. So the sample is tilted under the electron beam, and the image is acquired in order to retrieve the information in 3 dimensions. But why is it so important to get this 3D information in tomography? Is that because, for instance, if we only have 2D projections, it can actually hide the truth behind the true assembly of the object. So as illustrated on this nice piece of art, you have here a 2D projection of a man, so in other terms, a shadow. But if we dive into the truth, you will see that this shadow is actually made of different parts that don't look like a man at all. And this is just an illustration to show that projections actually do not show the full assembly. And that's how we can benefit from 3D information and tomography. So we managed to obtain high-resolution images of the spores. At that time, I was mainly focusing on ultrastructure, meaning that the pixel size was quite big, so I could have a full overview of the spore. And for the first time, we obtained really nice high-resolution images of the cellular content. So to facilitate visualization, it's possible to do segmentation. So it allows to have an idea and an easier idea of what's in the volume. So I could segment the mother cell envelope with the cell wall and the membrane. I also segmented the spore envelope that has 2 membranes in peak, separated by a thin cell wall layer that I represented here in blue. It is possible to segment the ribosomes. So here the segmentation was powerful enough to sometimes distinguish the 2 subunits in the bacterial ribosome. I also segmented the coat proteins, which are important for bacterial resistance, and other features such as the polysaccharide storage granule, the flagellum, and many more. So segmentation provides a nice overview of the cellular content and illustrate the power of cryocellular tomography. I would like now, before going to the result, to emphasize on the concept of mining the tomogram. So actually, this tomography provides kind of a snapshot of the cellular content, of the full cellular content in a 200 nanometer fixed section, meaning that there will be probably a lot of buried information, and it is crucial to keep an open mind, to keep an open eye, because there is probably much more present in these tomograms than what was initially expected. And therefore, I think that in situ cryotomography really provides a novel way to explore the cellular content. So going back to the spore, I said it was a resistant kind of a cell, but it can be seen actually as the extremely resistant survival form of bacteria, because it resists a wide range of stresses such as extreme pH, extreme temperature, most bleach-free disinfectant, radiation, and because they are dormant, which means metabolically inactive, they are inert to antibiotics. So this super resistant behavior is possible thanks to several resistant mechanisms, such as the protection of DNA by small acid-soluble proteins that compact the DNA in a quasi-crystalline structure in the spore core, the presence of dipicolinic acid that dehydrates the spore, the cortex, which is the cortex and the coat, which are 2 external protective layers. So the cortex is a modified type of cell wall of peptidoglycan, and the coat is a multi-protein layer on the outer part of the cell. And as I mentioned, metabolic shutdown, which make the spore inert to antibiotics. So today I will focus mainly on 2 aspects of this resistance, which are the DNA protection, compaction, and the formation of the coat. So the goal of my PhD was to provide data, structural data that will allow and help to understand the assembly of those structures. So we first started to see those kind of filamentous structure inside the spore core, which correspond to the DNA. the DNA seems to form those filaments that then turn into toroidal structure. These toroids form at after 3 or 4 hours after the beginning of sporulation. And what's funny in this case is that you sometimes section the spore in another direction, so orthogonal to the toroid, and you would have the two pieces of the toroid of the DNA donut. And we think that these toroids and these filaments, it's a preliminary stage before the quasi-crystalline structure of the DNA that we see in mature spore. And this would be a step before full compaction. So going back to the coat, so the coat are those protein layers that are present on the outer part of the spore. It's many different layers, and it has been extensively studied, but mainly in mature spore. So assembly of the coat takes about 7 hours and involve more than 80 proteins. And this is huge because you have to consider that the division time of bacillus is about 30 to 40 minutes. So it's a huge commitment to spend 7 hours to really make this coat. Among those 80 proteins, only 10 of them show a phenotype when deleted, and these have been named the morphogenetic proteins because they show a strong phenotype. So previous study of the coat were mainly either low resolution imaging, fluorescence imaging, or were focusing on late stages and mature spore. So for instance, it was thought for a long time that the coat was an amorphous structure until late stage of sporulation. During my project, we decided to investigate early coat assembly, early stages of coat assembly, because in the frame of developing antimicrobial compounds, for instance, it is important to decipher the structural step before this protective layer become impermeable. So at early stages, it's still accessible to external molecular agent. And what our data revealed is that the coat actually display the early coats display 5 structured sublayers. So let's go through them. So because we managed to take images at different time points during sporulation, we were able kind of to follow the time course of assembly. So at the beginning of sporulation, 1.5 hours after the onset of sporulation, the asymmetric septum starts to bend due to DNA entry, and we start to see a crenellated structure at the surface. So small dot that starts assembling at the spore surface on the mother cell side. A bit later during sporulation, when the mother cell starts to completely engulf the spore, we start to see appearing a white amorphous matrix that is capped by a thick matrix and itself covered by bead-like elements on the outer part. Even later in sporulation, when the spore is fully contained into the mature spore, into the mother cell, sorry, we see a bit better. So the layer starts to organize even more with the light matrix, then the dark matrix, and the bead-like elements that really seems to cover the outer part. Even later, we start to see a final layer, which is much denser and more localized somehow, and this we call that the dark smooth layer. So segmentation allows to again visualize the different layers and their position. So in green you would have the thick dark matrix and the different bead-like elements in orange and red, and the dark smooth layer that seems to be localized at the spore pool. So we took advantage of the 3D property of the spore in order of the 3D property of the tomogram, sorry, in order to dive into these different layers. And it is possible, actually, if you rotate the tomograms, to look at the surface of those layers instead of their side. So for instance, if we rotate the tomograms at the level of the 4-spore membrane, you will see here the 4-spore membrane from the surface. So you would have the outer 4-spore and the inner 4-spore that forms this black ring in the middle. And in between, you have this thin, darker layer, which is the modified cell wall layer that is known to be there, to be in this intermembrane space. So we did that for all layers, and we could really see a succession of densities that are distinct, strongly supporting the fact that we have a distinct organization, distinct layers in this formation of the coat. This all the way to the dark smooth layer, which is the denser of all. So we wonder what was the role of the morphogenetic proteins, so the one that have a strong phenotype when mutated. So we investigated several mutants, and here is an example. But for instance, if you delete one of these morphogenetic protein, you completely lose the bead-like element, so the orange and red that was represented. And you have directly the dark smooth layer that actually delocalize and starts to be on the side rather than on the pole of the spore. And at that, we could also see many spore defects depending on the mutants. So at that time, access to the film milling instruments was not that easy. So we decided to go for another technique, which is a freeze substitution, and to study even more mutants. And as a matter of comparison, here on the top, it's the freeze substituted sample, and on the bottom, it's the cryo-tomogram. And we could see similar organization. So in the resin, you have much less details, but we have also the clear matrix, the dark matrix, and the different upper layers. So we managed to produce a resin sample of many mutants, and comparing the defect seen in mutants to what was known in the literature regarding the interaction between the different morphogenetic proteins, we were able to propose a novel model for early coat assembly. And what this model brings is a precise measurements of the different layers and the distances between each putative morphogenetic proteins. So now, briefly before finishing this presentation, I would just like to say that I'm now at the University of Geneva, and I keep doing cryo-ET because I think it's a really great technique, but this time on yeast. And these budding yeast are quite a nice sample to work with as well. And we are now also kind of interested in compartments, but I'm more interested in some plasma membrane compartments that are seen in yeast and are known to be involved in stress response. So we are now, during my postdoc, investigating the formation of the structure, and we would like to understand the structural interplay between those membrane compartments and the plasma membrane homeostasis controlled by the kinase TORC2. So with this presentation, I hope I managed to show you that tomography, in situ cryotomography opens a lot of different projects, and I would like to thank all the people that were involved, my two PhD supervisors, Cécile and Christine, but also the members of the facilities, and of course, you for your attention.
Thank you very much, Elda. Your work is great. So maybe we can go for the question that Sarah has posted. So she says, "Hi Elda, you mentioned the Tokuyasu method at the start of your presentation. I haven't heard of this technique before. How does it differ from free SUF's Themovis?"
So Do you still see my screen? Yes.
Yes.
Okay, I can just show you some images. So, so freeze substitution. So I will just explain briefly what are the three of them. So freeze substitution is a way to you freeze your sample and in a vitrified state, and then you progressively remove the water and replace it to organic compounds in cryo conditions, and finally you embed your sample in resin. And Tokoyasu, some of it is kind of the same, meaning that you freeze your sample by high pressure freezing, so you get them in vitrified state, and then you directly section this vitrified material with a diamond knife. Tokoyasu is a different technique in the sense that the sample is embedded in cryoprotectants and gelatin, and it's not high pressure frozen. It's just frozen in liquid nitrogen. But because it has embedded in those sucrose and gelatin, it kind of preserves somehow the ultrastructure. So you see here on these three images that the ultrastructure in Tokoyasu is less preserved, meaning that the cell is a bit weirdly shaped and this kind of thing, but it really well preserved the antigens. And this is why it's interesting, because it's the best technique actually to do immunolabeling. And because the sections can be bring back to room temperature, and immunolabeling can be done on these peculiar sections. So it's a third technique, a kind of in-between technique.
Thank you, Elda. And now we have another one from Carmen San Martin. She says, great work, Elda. 2 questions. Are there any proteins involved in regulating genome compaction? And can you discuss your experience with cementation software? Thanks.
So there are those small acid-soluble proteins that are known to be produced at the beginning of sporulation only on the spore side. And they are produced in a high amount, and they are thought to interact directly with DNA. And it is known that in vitro, if you purify those proteins in presence of DNA, it will naturally form those spirals or higher order DNA compactions. So these small acid-soluble proteins would be the main driver of DNA compaction. Concerning segmentation, yes, so I've done sometimes manual segmentation when the neural networks, for instance, were not able to segment properly. But for the ribosomes and the very, when I have a lot of this, the thing I want to segment in the tomogram, I used EMAN2. EMAN2 has these neural networks integrated, and it's really powerful to actually segment your tomograms. So you just have to train. For instance, you select 10 ribosomes and you draw inside the ribosomes where is your ribosome, and then the neural network will be trained on that and is able after that to get all the ribosomes in all the tomograms that were acquired at the same parameters. So yes, my 2 things are iMod for manual segmentation and EMAN2 for automated segmentations.
Great, Elda. Thank you very much. We also have a couple of questions to Jirka, to the previous talk, but since we are out of time, I encourage Spencer, Wilhelm, and Karina to contact him directly. And well, nothing else. This session will be at some point available at the YouTube channel of Instruct. And I think that's all.